Hi everyone, Dr. Ron Bio here to discuss an important concept for AP Bio or any molecular biology or biotechnology class really, and that topic is restriction enzymes. So these enzymes are tools in the lab. They basically help researchers manipulate DNA. This particular video is going to be a beginner level video because we only discuss examples of linear DNA being digested by restriction enzymes. There are other Doc Ron Bio videos that discuss the more complicated examples, including plasmid digest. So let's go ahead and get started. I should start by saying a quick reminder that the molecule that we'll be manipulating in these reactions is DNA. So you have here the classic double helix that we've all come to love. Here I've shown our DNA straightened out into a ladder form. It's always easier to see the different strands when it's straightened out. However, we're going to show it as just a straight line. So I want to keep in mind what we're looking at here as we proceed through these videos. Okay, so restriction enzymes recognize palindromic sequences. So these are specific sequences of DNA inside the molecule. Restriction enzyme recognizes a specific sequence. And really, these enzymes have evolved in prokaryotic cells as a protective measure. You may recall that viruses called phages can use bacteria as hosts, and when that happens, viral DNA is inserted into the cell. When these enzymes, these restriction enzymes can chop up the DNA and prevent the viruses from using the cell as essentially a viral incubator. So restriction enzymes are protective in nature. They recognize these very specific sequences. Scientists have been clever enough to use these restriction enzymes and their ability to recognize specific sequences in the lab to manipulate DNA. Okay. So each restriction enzyme is essentially named after the organism that it was discovered in. So I'm going to show you how a certain restriction enzyme called EcoR1 works in this example. So again, E. coli is the name of the organism that this one was discovered in. So we get EcoR1. EcoR1 recognizes the sequence GAATTC, and you can see it on both sides of the molecule here, proceeding in either direction, GAATTC. Okay, so that's a palindromic sequence specific to EcoR1. The enzyme catalyzes the breaking of the phosphodiester bonds in the sugar phosphate backbone of the DNA molecule. So you can see that phosphodiester bond right here being broken by EcoR1 and right here being broken by EcoR1. So these bonds are broken. The molecule is said to have been digested. You can see that the way the molecule is digested creates sticky overhangs in either segment of DNA on either side of the break. So we have here a sticky end overhang over here and a sticky end overhang over here due to the break in the phosphodiester bond here and the phosphodiester bond here. We've also broken away some weak hydrogen bonds between the DNA molecules. So keep in mind that A and T used to hydrogen bond, A and T used to hydrogen bond, T and A used to hydrogen bond, and T and A used to hydrogen bond. So we've broken the phosphodiester bonds, but we've also gotten rid of the H hydrogen bonds here that are weaker in nature. Okay, so this is really useful for cloning purposes, but we're not going to get into that exactly here. So let me show you the weak hydrogen bonds that we've broken. There are, of course, other restriction enzymes that recognize different palindromic sequences. So here's an example of HIN3 and PST1, where you'd get breaks here, and then removal of these hydrogen bonds and breaks here and here in this particular restriction enzyme. So let's do a few examples here to see what happens in these reactions in the actual DNA molecule. The following examples, I will draw a line to indicate the DNA, as we said before, but keep in mind we are actually dealing with a double helix, or however you imagine it, a straightened out ladder of DNA. Okay, so example 1 is a single digest of a 20 kilobase fragment of DNA with the restriction enzyme EcoR1. You can see from the figure that this restriction site is dead center in the molecule. Therefore, we will have 2 fragments when we perform this digest. So if you take a 20 kb fragment, you will get one 10 kb fragment here and one 10 kb fragment here. Okay. There are computer programs out there that actually help you determine the size of your fragments down to the base, but just for general learning purposes, if you take a 10 kb length of DNA and you have a digest restriction site in the middle, you get 2 10 kb fragments. The second example is also a single digest. Here we're going to digest with a different restriction enzyme called BAMH1. The restriction site is clearly not in the middle, so you have to estimate to make your prediction. So again, if this is roughly the middle of the DNA molecule, I'm saying that if all of this is 50% of the molecule, that this is roughly 25 and 25%. Okay, so by my estimation, we would have a digest that would lead to roughly 25% of this 20 kb fragment over here. And then we don't have a restriction site there, we're just approximating, and then you'd have a fragment about this length. So our new fragments would be 25% of 20 kb, so there'd be a 5 kb fragment, and then the rest would be the other 75%, which represents 15 kilobases. So you'd have these 2 very different size fragments in this second example. Let's move on to a third example. This will be our final example. It's a double digest. So here we're looking at a digest in which you have both EcoR1 restriction enzyme and BAMH1 restriction enzyme. So if both fragments or both enzymes are working, you actually create 3 fragments. This fragment here, this fragment here, and this fragment here. This fragment is about half of the 20 kb, so that'd be a 10 kb fragment. And you have 2 halves of the half, so this would be a 5 kb fragment, and this would be a 5 kb fragment as well. So three fragments there, but only 2 different sizes. Now this is very basic. There are other complicated videos out there that I suggest you check out. So let me know if this helps. Let me know what you think of the video. Once you get this level down, check out the other videos that will involve circular plasmids or the application of gel electrophoresis. Okay, talk to you next time.